have a short time at the end for questioning. So next up is Dr. Zave Kain. He's the current chair of the Department of Anesthesiology at UC Irvine. Just happened to be also an NIH-funded investigator and an international expert in the pain management and various psychological issues dealing with pain management in children. And so we're very happy to have Dr. Kain here. He's also Associate Dean for Clinical Research in the School of Medicine. Dr. Kain.
Thank you and good morning. I am the last speaker before the panel, before the break. So I promise to be 15 to 18 minutes because otherwise people will start leaving. Over this period of time, we're going to talk about some underlying conceptual framework regarding clinical trial and the pharma industry. I was afraid of this. Okay. Disclosure. Can't start it without it. I've received pharma support from Hoffman, LaRoche, and Nesta and Aspect. The last one was in 2001. I've been an NIH investigator since 1996. And since I'm going to give you a piece of my mind about the editorial boards, I do have to disclose that I'm a member of all five, well, not all five of them, 5 out of a million, but of 5 of them. So before we start talking about the pharmaceutical industry and clinical trials, let's look at this one. Between 1981 and 1990, 196 new drugs have been approved by the FDA. 92% of them were developed by the pharma industry, which, by the way, has a budget of $6 billion. The budget of the NIH, to compare it, is $29.2 billion. There's no question the pharma industry advanced significantly on scientific knowledge. It increased our lifespan, and certainly they give some dollars for treatment of diseases in developing countries. And by the way, did I say that they have a huge research budget? Because they do. But the pharma industry has another side to it. And I don't know if it's the right cat or the left cat, but one of them is the next side. And the next side is a bedside story. So I'm a pediatric anesthesiologist by training. We put 21 million people in this country under general anesthesia every year. One of the not common complications, I have some surgeons in the audience, is awareness. What is awareness? Awareness is when you induce general anesthesia, you think somebody's under general anesthesia, the surgeons are operating, but guess what? The patient is actually awake, but they're paralyzed. So they're paralyzed and awake, they may or may not feel pain, they cannot tell you that they are awake. That doesn't sound good, right? So it's not a good thing. It does cause significant psychological problems, immediate and delayed. What are the solutions? There are really not great solutions out there. The old solutions are looking at heart rate and blood pressure. The new solutions is to look at something called BIS, bispectral index, spectral index. Now, this is BIS. The global market leader for BIS is Aspect from Boston, which relies on a proprietary algorithm for processing an EEG and alerts anesthesiologists the depth of anesthesia inadequate. This is the independent device. This is how a screen looks in our operating room. The last one, 70, is the BIS. The BIS has to be 40 to 60 to be general anesthesia. Above 85, the patient is awake. Very simple, right? Really, really simple. Compressed EEG. That's wonderful. And so Aspect was very smart. They're amazing people. They have sponsored literally hundreds and hundreds of articles that were published in every journal you can imagine that show that if you put BIS on a patient, there is less awareness issues. And by the way, you need less anesthetics. And by the way, the patients wake up earlier so you can do more surgery. And then came an article by the New England Journal of Medicine. Now, this article was published in March. It came from WashU. This article did not get any money from Aspect, one of the only studies out there. In fact, the only large-scale study out there that did not have pharmaceutical support. So what did they find? No reproduction of the results of previous studies that reported lower anesthesia awareness with BIS monitoring. Wow, okay, but there's 786 that said that there is. Well, they didn't find it. Awareness occurred even when these values were within target ranges, and findings do not support routine BIS monitoring as part of standard anesthesia care. As a practicing anesthesiologist, I can tell you, we have seen this a year ago or 2 years ago. In our practice, there was a problem, but the data out there said, no, it's really a good stuff. So what happened next? This is from Google Finance. This is the value of this stock. This is when the New England Journal article was published. Look what happened to the stock. Dropped by 50%. Why am I putting this slide up there? Because this is one of the motives that there is to this entire industry. Well, is this case unique? Okay, so it's Aspect Technologies from Boston, Massachusetts. What does that mean? And this is from New Scientist. Research funded by drug companies is biased. This is from the San Francisco Chronicle. UCSF study questions drug trial results. When drug companies sponsor research, the products are more likely to perform. They actually looked at about 200 studies that looked at statins, and they showed that if the study was supported by drug companies, it's 20 times more favorable, had made more favorable results than if it wasn't supported. By the way, why do they do all these studies on statins? Well, because there are many, many, many statins, but they don't support pediatric cancer research because there are many more patients who are on statins, and there is no money in pediatric cancer research. And that young adolescent group, there are very few of them. And so the money is not there, and therefore we have the Me Too drugs, and the drugs that go to a population that is more likely to use them. Lisa Byrne, who was part of the other study, did this very interesting study, published in the British Medical Journal. And what they have done is they wanted to see if financial ties to one drug company are associated with favorable results in a meta-analysis of antihypertensive drugs. This time, all the studies published up to 2004 made outcomes where results and conclusions. And what did they find? 40% of all studies published to date, that's 2004, it's a total of 124 studies, had financial ties to a drug company. They showed in univariate analysis that if this study came from a drug company ties, it showed that the drug is better, but then they did logistic regression model in which they put other variables that may actually impact the outcome. And they found again that if one drug company remained more likely to report favorable conclusion. In fact, they also showed that 37% of the drug companies' articles had poor concurrences with the results and the conclusions, which mean, and one of the speakers showed it earlier, that if you look at the body of the article, it says, maybe it's effective. But if you look at the conclusion, they'll say, it's effective. What about other studies? Is this one case and one study that I'm showing you? Nope, multiple. This was published in the Lancet in 2000 on multiple myeloma. This was published at the Journal of General Internal Medicine. All these are meta-analysis of these various studies. The next one, trial drug companies support are more likely to report in favor of experimental therapy. Last one, only 5% of industry-supported analysis reported unfavorable conclusions compared to 38% of non-for-profit sponsored studies. So we certainly have an issue. So why is this happening? Well, one issue is the publication bias. And it's not only that we are more likely to publish positive studies, it's also that the drug companies actually controls the results of the studies, and therefore, if the results are not favorable, you may or may not, as an investigator, have the right to ever publish that paper. But here are some other things, some other ideas, and I deliberately did not make this slide of my own. I wanted you to see what's out there, what people are thinking. Conduct a trial of your drug against treatment known to be inferior. Okay? That's interesting. So you do have a randomized controlled trial, right? It is evidence-based medicine, but it's a question of how do you actually design the trial. How about this one? Trial your drug against another drug that's too low of a competitive drug. Or the other way around, conduct a trial of your drug against too high of a dose of a competitive drug, making the other drug or your drug look less toxic. Conduct a trial that is too small to show differences from a competitive drug. That's obviously type 2 error, right? Use multiple endpoints of a trial. Remember, p less than 0.05 means that 1 out of 20 cases you will have it by chance. Well, if you have 20 comparisons points, that means that you just guarantee that one of the outcomes will be positive. That's the game of statistics. Do multi-center trials and select publication results from centers that are favorable. And I can continue with this. But don't we have medical journals and don't we have editorial boards? Surely that's our job, editorial boards members, to make sure that these are never published, that we scrutinize the data. So this is interesting. This is from the former editor of the British Medical Journal, which, by the way, has been a leader in this area. And he published this very interesting article in the Journal of Royal Society of Medicine. And he talked about lapses at the New England Journal of Medicine. What the heck are they talking about? Let's talk about the recent editorial history of the New England Journal of Medicine. Do you guys remember this guy? Excellent editor. Was let go because, if you may remember, he criticized President Clinton vis-a-vis issues that were with Monica Lewinsky. So the Massachusetts Medical Society has decided to let him go. I don't know if you remember her. An amazing editor that was very vocal critique of the pharmaceutical industry. So they let her go. She came out with this book, which I really just recommend to read. It's great reading. And a lot of the questions that have been raised out here are answered in her book. She's just an amazing writer. Now, so what did the Massachusetts Medical Society do once they fired Marcia Angel? They went on and they hired Jeff Drazen, who's an amazing scientist, but who also had financial connections with 21 drug companies between 1994 and 2000. By the way, I don't know if you know this, but the major finances inflow to the Massachusetts Medical Society is the New England Journal of Medicine, and the major money there is drug advertisements. I'm not saying it's affecting it, just saying that we have to think about it. Let's talk a second about Vioxx here. This was published in the Wall Street Journal in 2006. The New England Journal missed the Vioxx warning signs, and it tells a story. In 2001, a Seattle pharmacist called a radio show in which Dr. Drazen, the top editor of the New England Journal of Medicine, was appearing, and she begged him to update an article that touted Vioxx while playing down its heart risk. Dr. Drazen was dismissive. We cannot be in the business of policing every bit of data we put out there, he said. He is the editor of the New England Journal of Medicine. We all know how that ended, right? 3 years later, they put it off the market, and an estimated 20 million Americans took Vioxx. There was a letter to the editor that was submitted at 2001 about this death. Dr. Drazen has rejected that letter to the editor. And by the way, the New England Journal of Medicine started to deal with their own data after a consultation with a PR consultant team that told them, well, maybe you should try and do some damage control. So there goes the New England Journal of Medicine. So it's not a shocking surprise that this was published in the New York Times about 2006. For science as gatekeepers, a credibility gap, or that the editor of Lancet said journals have developed into information laundering operation of the pharmaceutical industry, or that the medical journals are an extension of the marketing arms of the pharmaceutical companies. I'm sorry it's so sobering, but I wanted to be a bit controversial here. Or, this is an editorial from Neurology, how skeptical should we be about industry supporting studies? Okay, so how are we going to fix this? JAMA has started, and JAMA has come up with a fairly strict editorial policy. They have been real leaders in this area, and Catherine DeAngelis is just wonderful. Now, this particular editorial, which was published in 2007, was very interesting. They now require from studies that are supported by the drug industry to have a statistical consultant from academia. Well, what does that mean? Well, no. Now it means that you actually have to expose your data to somebody from the outside. Well, how did the pharmaceutical industry respond to this? Not good. So here is an article that was published in the British Medical Journal, and the year was 2005, by the way, for the other article. The author of this article, one of them was from the pharmaceutical industry, and he said, extra scrutiny for industry-funded trials, JAMA demand is unfair and absurd. And they compared it the same as they say, well, you know, it's the same if you ask now authors from Western Europe, for example, to have a statistical editor. What the heck does that mean, and why should studies that come from industry be different? Well, they should. And so let's look at some issues. Appropriate controls. When you deal with studies like that, you have to deal with control dosing issues. So when you compare, it's not enough to have a randomized controlled trial, right? Look at the control group. Make sure that the control group is the gold standard at the current time of treatment of that disease. The gold standard. You better make sure that it's the right drug at the right dose when you compare it against. You better make sure that you have the appropriate sample size, right? Because if it's too small, you will prove that there is no difference between the two groups. That's type 2 error. External validity. This is one of the biggest issues that there are out there. A lot of pharma studies take a select group of patients, healthy young adults, and do the studies on them. They do not do studies on young children. They do not do studies on fragile elderly people. Now, why do they do that? Well, A, it costs a lot of money. B, it costs a lot of money. And when you do a PK/PD studies, if you do it for an adult, you need to do 1. If you do it for a child, you need to do about 20 because their pharmacokinetics changes. So you need to do it for the ages of up to 1 year, 1 to 3, 3 to 5, 5 to 7. So we have a problem. Surrogate endpoints. We saw it in the orthopedic talk that we have series of them earlier today. It really depends what outcomes you're checking, right? So if you developed a new instrument and you're looking at the patient a year later, before the cement had a chance to do anything, then you will show that you have great outcome. Okay, but maybe you should think about it ahead of time and choose a reasonable outcome. Clinically relevant, and that's something that we should always think about designing our studies is, when you look at statins, do I really care that statin reduce the level of cholesterol? What I really care is that statins have A, decreased the incidence of myocardial infarction, or B, have prolonged your life. That's what you really care. You don't care about the number, but it's much easier to measure level of cholesterol than look if you actually lived longer. Academic CROs, and I have to say on this one, the academic center have to take a big part of the blame on them. The reality is that our clinical research in universities have become more and more cumbersome and slower. We don't leave much of a choice to the pharmaceutical industry today, but to go to the private sector, but to leave the United States. And the result is then that it falls into the hands of individuals who get $2,000 or $60,000 per patient. And now they're really into revenue generating out there in Romania or, not to single out any specific country or India, but that's the realities of clinical research today. You have to have tight control of data analysis and interpretation by the commercial sponsor. Is undesired, and you have to give it to somebody from the outside. Let me show you one example of that. I'm sure you have seen this ad. Okay, this is for Lipitor, and this is obviously Dr. Robert Jarvik. You also hear it a lot. Reduces the risk of heart attack by 36%. Wonderful, right? But there's a small asterisk here. It says, that means in a large clinical study, 3% of patients taking the sugar pill or placebo had a heart attack as compared to 2% who had taken Lipitor. So we're talking about a relative risk reduction versus an absolute risk reduction. Now, just look at the way the ad is. Isn't it pretty amazing? Black, white, beautiful, big number, small, no difference, and it's right there. Okay, let's look at their data right now. So this is a traditional survival curve. You have the cumulative incidence, and you have the number of years. And if you look, let's look at this one. The absolute risk reduction at 3.5 years down the line is 1%, right here. So it's the delta between 3 and 2. And on the other hand, if you look at And these numbers, you can see that the hazard ratio, which is very similar to risk ratio, it's essentially almost the same, it is 36%. Okay? But how should we have presented this data? There is a small known statistics. It's called number needed to treat. What is number needed to treat? Actually, how many patients you need to treat in order to see a benefit. This particular study has to be 1 to 100. What does that mean? You take 100 patients. You treat them for 3 years with Lipitrol. 1 out of that 100 would have prevented one NMI. 1. 1 out of 100 over 3 years. That's the statistics that presented in this article. Now, this is an interesting one. This was presented at the Business Week at about 2007, and it looked at the number needed to treat and how it changes. So 1.1 is antibiotic cocktail. So obviously, you need to treat one patient, actually a bit more than one, in order for the drug to be effective. It's effective for the first patient. Let's look at this. Zetla, lower cholesterol, 1,000. You need to treat 1,000 patients, in this case over 5 years, to show that actually it does anything. And by the way, this drug doesn't even do anything for heart attacks. Look at Lipitrol, it's between 70 and 250. What does that actually mean? Let's look at this one. So NNT of 250. What if you put 250 people in a room and told them that they would pay each $1,000 a year for a drug that they would have to take every day, that many would get diarrhea and muscle pain, and that 249 would have no benefits, and that they could just do as well by exercising? How many of them will take that? That's a pretty controversial question, right? And in fact, those 249 people, they actually have problems, right? Pfizer have been sued over Lipitrol because of issues related to memory loss, nervous system damage, and so on and so forth. So it is a controversial issue. We cannot simply let out of our control the data that are being presented in journals by pharma-supported studies. We have to have some kind of access to that data. Otherwise, we end up with things like this. I will spend 1 minute on this, and this is one of my pet peeves. This was published last year. It's called The Legend of the P-Value. And essentially, for the physicians of the audience, physicians go by P-value. If it's less than 0.05, we basically say, okay, the drug is good. And it's not the case. P-values can be manipulated by a sample size. You give me a sample size large enough, I'll show you anything you want under the sun. And so in this editorial, what I'm advocating is that besides looking at the sample size, you really have to look at how large the difference between the primary endpoint of the group is, which is what is the effect size. And the effect size is much more statistically significant because not at the P less than 0.5 level, but are the differences statistically significant. And then you have to look at clinical significance. If you reduce cholesterol from 240 to 220, I will show it statistically significant because all I need to do is recruit 10,000 patients to this study, which most pharma studies do. But clinically, does it matter? Those 20 milligram percent do not matter at all. And nobody talks about it in our published literature, which is a huge issue. We have to have independent data in safety monitoring committee. We just have to have it because the fact that the pharma industry does not let you look at the data, again, is a problem to me. Publication issues and control, again, when you're an investigator who works on a study supported by a pharma company, there has to be an agreement ahead of time that the results will be published regardless of anything. And it's not the case. You will see in the small letters, it will tell you that they control the publications. And last but not least, you obviously has to disclose all your interests, as multiple speakers have indicated before. So to conclude all this together, we have a myth that says that you should believe everything you read in a medical journal, and the reality is that life is not that simple. Thank you.
like to thank all the speakers on the panel. And next up, Dr. Rosen is going to have a few words.
I think for the sake of time, we'll have the Q&A session for everyone at the end. And I think now we'll take a break and be back here at 10:10. 15 minutes.